Hello everyone. Today I'll be presenting on ABO blood grouping, one of the most important laboratory procedures in immunology, and it's performed in hematology and transfusion medicine. Blood grouping helps to determine an individual's blood type and ensures safe blood transfusion. During this presentation, I'll be explaining about ABO blood grouping and RH systems typing and the testing procedures of the same and interpretation of the results and clinical application of the same. So let us move to the next slide. This slide introduces about Karl Landsteiner, who is known as a father of blood grouping. In 1901, Landsteiner discovered the ABO blood group system by observing that mixing blood from a different individual sometimes resulted in clumping. This is known as agglutination, and this discovery revolutionized blood transfusion practices by, by making transfusion much safer. Later, together with Alexander Wiener, he also contributed to the discovery of RH system, RH typing. For his groundbreaking work, Karl Landsteiner received a Nobel Prize in Physiology in Medicine in 1930. And we need to know about the ABO blood grouping system. What are the types distributed over there? And this talks about, this slide is talking about different types of blood groups. So ABO blood group system classifies into four types based on the presence of A and B antigens on the surface of the red blood cells. So red blood cells are distributed with either anti-A or anti-B. I mean antigen A or antigen B, if in case if it is A blood group or B blood group. If it is distributed with both, it is AB blood group. If there is no antigen, then that is O blood group. The surface is distributed with the D antigen also, that is determining the RH typing. So the RBC will be interacting with the other and it will be generating agglutination or clumping. So blood group A possesses A antigen, B possesses B antigen, blood group AB possesses both A and B antigens. Blood group O lacks both A and B antigens. So individuals naturally produce antibodies against antigen which they do not possess. This antigen-antibody interactions, which is forming the basis of blood grouping, and this interaction will result in a clumping that is nothing but agglutination. That is what we are going to look into in the remaining slides. So this is a slide which is representing the major components of the blood. It is consisting of 55% of plasma, 45% of formed elements, and it mainly contains water, protein, and electrolytes. I mean, the plasma contains water, proteins, and electrolytes. The cellular components include red blood cells, white blood cells, and platelets. Among this, red blood cells are important for blood grouping because they carry A and B antigens and also the D antigens, which is responsible for RH typing on their cell surface. So this is a figure which is summarizing about the four ABO groups. Blood group A has A antigen and B antibodies, B has B antigen and A antibodies, and AB has both the antigens and do not have A and B antibodies. And O has neither antigen but possesses both A and B antibodies. This classification is essential for compatibility of blood donors and the recipients. This is a compatibility chart which helps us to understand which blood groups can donate or receive blood safely. For example, O negative is considered as a universal donor because it lacks A antigen, B antigen, and also RH antigen, that is D antigens. Group AB positive is an universal recipient and it has all the antigens, does not produce antibodies against A or B. So matching donor and the recipient groups prevents a serious transfusion reaction. Moving to the next slide. So what are the materials required for performing blood grouping? And it includes basically we need a glass slide, a sterile lancet, and spirits, and we need anti-sera for anti-A, anti-B, and anti-D. If we want to move with the A subtypes, then we need A1 anti-sera also. Then for wiping, we need cotton and mixing sticks after addition of this anti-sera into the blood. And we need a disposable gloves. Each reagents over here plays a specific role in identifying the corresponding blood group antigens. What are the precautions to be followed in blood grouping? Always use sterile lancet to prevent infections. Wear gloves throughout the procedure. Clearly label the slide before adding the reagents. Use separate mixing sticks for each reagent to avoid contamination. These precautions ensures accurate and reliable results. There are two commonly used methods for blood grouping. Either we can go with a tube test or a rapid slide test. The procedures we are going to look into. Both the methods are based on the same principles, antibody antigen interaction. The choice of method depends on laboratory requirements and the level of accuracy needed. The ABO blood group test tube test is a manual, a gold standard laboratory method used to accurately determine the exact blood type either A or B or AB or O, and also the RH blood group either positive or negative. It involves two complementary parts, that is, forward grouping, test your red blood cells, reverse grouping is testing your serum or plasma. How the tube method works, we need to know by using separations, forward typing and reverse typing. So we need to separate it and through centrifuge, I mean separate the red blood cells from plasma and serum. We can go with the forward typing. For example, forward typing, the patient red blood cells are mixed with the commercially prepared anti-A or anti-B reagents in separate test tubes. If a blood clumps agglutinates, it means a corresponding antigen is present in your cells. So it says that whether A blood group or B blood group, wherever it reacts, it is named accordingly. In the reverse typing, the patient's serum, which consists of natural antibodies, is mixed with a known red cells, I mean a cell or a B cell, I mean A cell or a B cell. The clumping here conforms the opposite blood types, providing a vital cross check to verify the forward typing results. So here we can identify this slide is representing the identification of the modified tube test. So three test tubes are labeled as anti-A, anti-B and anti-D. One drop of corresponding anti-sera, I mean A, B and D will be added in each tube, and it can be added with the red blood cells or a serum or a plasma. So here one drop of it is added and mixed well. The tubes are centrifuged at 1000 rpm for a minute. Finally, the tubes are examined for agglutination. The presence of agglutination indicates the corresponding antigen is present. It says that whether it is A blood group or B blood group or O. If there is no agglutination observed in A and B, which is saying that it is O blood group. And if there is agglutination in the third tube, then it is positive. And if there is agglutination, then it is going to be, if there is no agglutination, it is going to be negative. So it says that the blood group is A, B or O. Either A, B will be there along with that. As it's shown in the previous slide, the agglutination reaction, the same reaction, it is going to be performed in a slide. This is nothing but a rapid slide test. 3 labeled areas are prepared on a clean slide. 1 drop of each anti-A, anti-B and anti-D reagents are added, to which 1 drop of a blood is mixed separately with each reagent using different mixing sticks. The slide is generally rocked for about 2 minutes, and visible clumping or agglutination indicates the positive reaction. What is agglutination first of all? Agglutination is a reaction in between the antibodies which we are adding and the RBC which contains antigens. So it is going to be an antigen antibody reaction. So the antigen is here is RBC, the antibody whatever we are adding, it is going to interact with that. Agglutination is a process of clumping of particles like red blood cells or a bacteria suspended in a liquid. In biology, it is an antigen-antibody reaction where specific antibodies bind to multiple antigens forming a visible cross-linked clumps. This is nothing but agglutination. It is widely used in clinical medicine for blood typing and rapid infection disease testing also. And here we could able to observe the agglutination reaction over here. And after performing the test, so we need to observe the reaction carefully. The agglutination indicates that the corresponding antigen is present on the red blood cells. No agglutination indicates that antigen is absent. And both the slide test and tube test are interpreted using this principle. Moving to the next slide. In this example, blood sample is showed no agglutination with anti-A and anti-B and agglutination with the anti-D. So what does it mean? There's no antigen presence of antigen A, antigen B, which represents it's an O blood group. And it is seen with the anti-D, I mean agglutination is observed in the anti-D against anti-D, which says that RH is positive for that. So we are supposed to interpret an individual as O positive because individuals lacks A and B antigen but possesses RHD antigen. ABO blood grouping is essential for clinical procedures like blood transfusion and organ transplantation to prevent the fatal immune reaction. So it has number of applications. It involves collecting the blood sample and performing two tests, either forward typing or reverse typing. I mean, the forward typing is talking about antigens, detecting the antigens of and the red blood cells, and the reverse typing is talking about detecting the antibodies in the plasma. The practical application and implication of determining ABO blood group has several fields. For example, blood transfusion. I mean, the transfusion medicine, which determines the blood products you can, whatever you receive safely. For example, O negative is universal and blood donor, and AB positive is a universal recipient. And maternity screening identifies maternal fetal blood incompatibilities like RH incompatibility, allowing preventive treatments during the pregnancy. And forensics, looking at the forensics, it assists law enforcement in analyzing crime scene evidence and narrowing down suspect profiles. So paternity and legal cases it also has application. It is used for analyzing the genetics, I mean genetic testing and ancestral population studies to establish biological relationships. To safely track and verify your own blood group, you should refer to the documentation provided by certified diagnostic centers or hospital records. You can also utilize authorized online health products to view your medical history. If you like to read more specific compatibility rules, and you have to look at the number of books available and diagnostics tools available, and which says that compatibility testing is more important with respect to transfusion and also with respect to paternity and legal cases.